Okay, so today's subject is going to be another cheerful neurological condition, Alzheimer's disease. And there is some debate as to what extent it is different from some of the processes that occur during aging. So in normal aging, you obviously have like large numbers of changes throughout your body, usually not, at least once you pass sort of your 20s, the changes aren't generally for the better. But parts of the, so in normal aging, you get parts of the brain shrinking. In particular, the prefrontal cortex and the hippocampus. And so those are important areas for, cognition and memory. You also get changes in the sort of white matter tracks that are connecting different bits of the brain together. You start getting changes in the blood vessels in the brain. There's a sort of reduced rate of flow of blood, and you sort of get less sort of growth of new capillaries. So bits of the brain gradually, just have their perfusion slowly reduced. You also get this buildup of, tiny sort of insults to the brain. So there's more and more damage by sort of free radicals that are generated by the normal metabolism. There's sort of like little increases in sort of inflammation throughout the brain. And sort of, none of those things are good things, but they all sort of seem to be just a function of like age. And although there's, is some debate about this, this is why it's sort of extremely unlikely, anyone would ever live to say 150 years old or something like that. In Alzheimer's, however, all these normal aging processes, even though they suck, in Alzheimer's, they are emphasized. They sort of occur at a more rapid rate. And that's really what gives rise to the sort of underlying symptomology of Alzheimer's disease. And so we're going to talk about today and tomorrow three of these sort of diseases which are particularly associated with aging and have particular cellular features as well. So all three, Alzheimer's disease, Huntington's disease, and Parkinson's disease, all show extensive neuronal loss. And if that loss is in regions that play a major role in cognition, then that will frequently lead to dementia. If the neuronal loss is in areas involved in control of movement, then that will lead to movement disorders, in particular sort of like Parkinson's disease. And dementia is just this progressive decline in cognitive function due to damage or disease in the brain beyond what would be expected from normal aging. So what does it look like? In Alzheimer's, this is sort of example of a post-mortem brain from an otherwise healthy individual and one suffering from Alzheimer's, who had suffered from Alzheimer's disease. And hopefully, yes, they should be age matched, yes. I did not prepare the specimens, so I'll trust the people who did it, but yes, they're supposed to be age matched. And so, like just at a glance, you can see that they don't look the same, right? Because they're sectioned through the same equivalent bit of the brain. And so this is sort of nice and sort of plump, if you like, and this is sort of all shriveled up. And so that's what's the changes that are occurring in the brain. This would be sort of very late stage Alzheimer's. But what you see is the cortex shriveling up. And so these, if you look at the gray matter here, it's noticeably thinner than it is in the otherwise healthy brain. And then the ventricles become much larger as well, because there's sort of more and more empty space in the skull to fill. And there's a severe reduction in size of the hippocampus. So essentially, they're losing a large amount of neuronal volume within the brain. So as far as symptoms go, the most striking early symptom is going to be memory lapses. usually this is a kind of minor forgetfulness, like, like where did I put my car keys, that kind of thing. But they get more pronounced as the illness progresses. And they also tend to favor older memories rather than newer memories. So you might still remember things that occurred when you were a child, but not remember, like where you spent last Christmas, for example. In the early and middle phases, patients are usually alert and responsive. But as it develops, they start having issues with some simple words or use of common objects. Then as it progresses, you start seeing more and more cognitive impairment, leading to sort of problems with language. So aphasia, skilled movement, apraxia, and recognition, agnosia. And also things like decision-making and planning. In the very late stages, patients will frequently be completely mute and completely sort of amnesic, as in, cannot really remember anything. In some patients are still sort of fully mobile, so that they can, kind of wander, but like it's without purpose, because, like they're incapable at that point of really planning. And so there's this inevitable, as the disease progresses, there's this an inevitable loss of the ability to, loss of judgment and reasoning. In sort of the late middle stages, patients will frequently develop delusions. About 10% of Alzheimer's patients develop Capgras syndrome, which is where they believe that their caregiver has been sort of kidnapped and replaced by an imposter. And so with this kind of thing, they're losing the judgment, and so they develop social disinhibition, and frequently coupled with uncharacteristic belligerence. And even in the same patient, though, this kind of ebbs and flows, so that they'll also have periods of extreme passivity. They have disrupted sleep-wake patterns, also, like down to the loss of neuronal volume, and so they start sort of like developing sort of nighttime wandering and things like that. Many patients will also develop Parkinson-like symptoms. So like a shuffling gait and generalized muscle rigidity and so on. But they don't have many of the other canonical features of Parkinson's disease, like, like resting tremor and intention tremors and so on. But there is a big overlap between the populations of suffering from Parkinson's and suffering from Alzheimer's disease. So this pronounced loss of neuronal volume is driven by apoptosis. So it's not like excitotoxicity like we were talking about with ALS. It's, like true apoptotic cell death, which means that the sort of neurons are individually just dying off in a nice sort of clean fashion, so they're not generating, bad, brain swelling or inflammation. However, when you're starting to see this sort of major decline in the population of neurons, then obviously that's going to lead to all of the symptoms that you see in Alzheimer's disease. And in Alzheimer's disease, well, in general, neuronal apoptosis can be induced by many different things. So immune cells, which would be, microglia in the brain, are, capable of essentially telling a particular cell to, like go apoptose itself. Protein misfolding and aggregation is a common cue for cellular apoptosis. DNA damage will also lead to that. Problems with energy metabolism will also lead to that. And oxidative damage will lead to actually problems with proteins, which will also trigger apoptosis. And many of those features are seen in Alzheimer's disease. The other thing that is commonly seen in Alzheimer's disease, and was actually in the very first report of Alzheimer's disease in, I think it was 1907, is the presence of these plaques and tangles. So, and these are tiny aggregations of proteins. And so the plaques, which are these sort of larger structures, are found, here's a bigger, a zoomed in picture of an individual amyloid plaque. So these are found outside of neurons, and they're big aggregates of amyloid protein. And the tangles are aggregates of a different protein, tau, and they're found within the neurons. So we're getting two different types of protein aggregate, one outside of the neurons and one inside. And they are seen to, they sort of slowly develop in sort of normal aging, but they accelerate at a greatly increased rate in patients with Alzheimer's disease. So if you took this, the hypothetical 150-year-old person and looked at their brain, they would also be probably chock full of plaques and tangles. However, you know, like no one gets to that point. So in an Alzheimer's patient, you know, like they might be 60 and have this huge buildup of these abnormal proteins. So the amyloid plaques, they're found outside of the neurons in the sort of interstitial space, and they're composed primarily of beta amyloid. And in Alzheimer's, they sort of tend to sort of have a particular pattern of their expression, which is different to what you see in normal aging. And it's still, the big issue is, well, actually, which is something we'll come back to right at the end of today's class, is sort of, you know, like what role do amyloid plaques play in the pathogenesis of Alzheimer's disease? In that initial description in 1907, there was Alois Alzheimer basically speculated that yes, the brains had these pathological features, but they were probably a marker of some other process that was occurring that was leading to them. And there is, you know, like there are now some clinical trials where they're really trying to get rid of these amyloid plaques to see if that is actually curative. And so if it is curative, then we know that they obviously are a fundamental part of the pathogenesis. If they're not curative, you know, you can pull away all of the amyloid plaques and have, but the disease is unchanged, then obviously it might be more of a marker rather than, you know, like a sort of side product of some other pathological process. So the second of these protein aggregates is the tangles, and these are formed from the protein tau. And we will pause there, and I have the box of terror for afterwards. Yeah. We will definitely talk about that. There is a little bit of genetic predisposition, especially with the AQE4. Well, okay. Who wants to be in charge of? You got. I'll offer this slide. Okay. So. Yep. What's it? You so say one of the key things that struck you, say for the first, or that you just talked about.
Like in late stages, Alzheimer's, Parkinson's, patients have some of the similar symptoms.
Yes, and that's why do you think that would be?
Because same areas of the brain like are affected. Makes sense that's similar.
Yes. Yes, perfect. And we do one more. This way, hopefully we'll get through everyone. Yeah, and accelerated. Yes, yes, exactly. Okay, and so the key question with Alzheimer's disease is, so I mentioned that the neurons are dying through programmed cell death, apoptosis. So the question is, why do they die? And which is, like really the question is, what is the fundamental disease mechanism in Alzheimer's? And so there are four different, hypotheses for the pathology of Alzheimer's disease. So there's this cholinergic hypothesis, amyloid hypothesis, tau hypothesis, and a metabolic hypothesis. And as we'll talk about each in turn, and the sort of thing to keep in mind when we're talking about them is, are they really telling us anything about the disease mechanism, or are they talking about how, are they more related to how the disease is causing symptomatic manifestations? So the first of these mechanistic hypotheses relates to cholinergic neurotransmission. So cholinergic neurotransmission is neurotransmission using acetylcholine as the neurotransmitter. So that's the same acetylcholine that you find at the neuromuscular junction. So this is the first of these hypotheses to be really developed. And it's proposing that Alzheimer's is essentially a specifically cholinergic deficit, and involving the cholinergic projections from a particular basal region of the basal forebrain. And this particular projection from the basal forebrain through the cortex, a cholinergic projection, was especially deficient in patients suffering from Alzheimer's disease. So, yes, everything kind of shrinks and dies and so on, but this particular cholinergic pathway was especially affected. Also, if you look sort of biochemically, you can see that the activity of choline acetyltransferase, which is an enzyme for the production of acetylcholine, was also found to be decreased in pathological samples from the brains of Alzheimer's patients. And a couple of other markers of cholinergic function in the brain were also downregulated. And this, in fact, is what has led to the first generation of anti-Alzheimer's disease medications. But this slide just illustrates this cholinergic projection. So there are some others, but this projection from the basal forebrain is a cholinergic projection that spreads throughout the cortex, and it's sort of like one little group of nuclei responsible for most of the cholinergic signaling within the cortex. The role of this is actually as a modulator of cognition, and it's thought to sort of like help refine information processing essentially. It also plays an important role in wakefulness, and so if you, for example, stimulate that portion of the brain, you can either elicit wakefulness or REM sleep, as distinct from like other stages of sleep. And if you inhibit that region, then it will actually induce deep sleep. And so for this reason, these cholinergic projections are very much implicated in studies of attention and also sleep. So that's like one possibility for what might be going on in Alzheimer's disease. You're messing with something that is like essential for organizing cognition. The second one we've already touched on is this amyloid hypothesis. So we know that we get these buildups of amyloid plaques. So are they the causative factor that's actually like ruining the brains? So in this way, Alzheimer's is often called a proteinopathy because it's like related to the misfolding and aggregation of proteins. Interestingly, the gene for the precursor protein for this beta amyloid, APP, is located on chromosome 21, and in Down syndrome patients have the duplication of that chromosome, which means that they have an extra copy of that gene. And typically, Down syndrome patients will exhibit many Alzheimer-like features relatively early in life. If you take this fragment of amyloid protein, the amyloid beta, it actually is like neurotoxic. And so people have generated transgenic mouse lines that sort of have an overabundance of amyloid beta, and they sort of show a buildup of amyloid plaques. And these specifically toxic fragment of amyloid beta has direct effects on synaptic transmission. The problem, which actually stems directly from these mouse models, but also from observation of human patients, is that there isn't a very good correlation between the sort of buildup of the plaques and the loss of neurons. And so it might be part of the puzzle, but it isn't necessarily the whole thing. In terms of how we generate these fragments, we start off with this APP, which is an integral membrane protein, which has a number of sites where it can be cleaved by endogenous enzymes. And in the so-called benign pathway, we have alpha secretase, which cleaves the protein, obviously at this point, and gives you this large soluble APP alpha protein. And that is thought to be an extracellular signal that promotes neuronal growth and survival. Obviously, since this is an integral membrane protein on the plasma membrane, any cleavage that's on this side is obviously going to release the protein into the extracellular space. If you cleave the protein in a different site, at this beta site, and then also cleave it with this gamma secretase site actually just within the membrane, you generate this A beta 42 protein. And the A beta 42 peptide is this toxic peptide that builds up in the plaques and seems to have some ability to sort of, so inhibit synaptic transmission and precipitate cell loss in some cases. And there's a theory that the amyloid plaques may actually represent the body's attempt to kind of diffuse the presence of these sort of toxic fragments by basically kind of lumping them together and sort of trapping them in a form where they can't do too much damage. There is also some kind of very indirect evidence which relates to a different protein, apolipoprotein E. And this is the sort of like one of the best characterized genetic aspects of Alzheimer's disease, because there are 3 alleles of it, ApoE2, ApoE3, and ApoE4. People who carry the ApoE4 allele have an increased risk of Alzheimer's disease. So however, about sort of nearly a third of the population of people who don't have Alzheimer's disease will have at least one of these E4 alleles. And about 2% of them are going to be homozygotic for the E4. Yes? So these are gene alleles. So that means that everyone has this protein, this gene, apolipoprotein E, but the different alleles are just different variants of that protein, or that gene that encodes the protein. It is in every cell, because these are all, like this is genetic, so it's part of your genetic code. So about 2% of people who have who never develop Alzheimer's disease will be homozygous for this ApoE4 protein, gene. And so what, like that, and they don't develop Alzheimer's disease. However, if you look at the Alzheimer's disease population, about a half of them have at least one of the E4 alleles. So there is this strong, there's a significant association, but it's clearly not causative. And being homozygous for the ApoE4 has a further increase in risk compared to just being monozygous. I think you're correct, but I don't really remember. I haven't looked at that for a while. Yes. So they're involved in cholesterol transport. So it's not, it's like just part of normal like cellular function, really. So, but obviously neurons are cells too, so they have like are dependent on many of these things. And neurons are obviously, or maybe not obviously, but neurons are in general the longest lived of all of our cells. So they might be more sensitive to some of these things, where, like if you think of a skin cell that might live for like, whatever it is, like 5-10 days, something like that. Like you will have neurons, or even I will have neurons that are, I've had my entire life. So in fact, the majority of mine. So, they are dramatically more long-lived than many of our other cells. So the significance of this is not so much that you need genetic counseling if you're a carrier of this. Yes? The length? Oh, linked. I don't know if there's a I don't know if there's any link with this with gender or not. So I mean, it's a good question. I don't know the answer. So anyway, this ApoE is associated with Alzheimer's disease, and its clinical value seems to be that it improves the certainty of Alzheimer's diagnosis. Like if a patient is presenting with early stages of dementia and they're a carrier, then you would be sort of more confident in giving that as the diagnosis. So what about tau? This is the protein that's in the protein aggregations within neurons. So Alzheimer's is also regarded as a tauopathy. It's part of a family of diseases where there is this presence of misfolded aggregates of tau protein. And tau is a microtubule associated protein. Normally, it's sort of like, it is regulated by phosphorylation. And in Alzheimer's patients, it becomes hyperphosphorylated. And hyperphosphorylated tau accumulates as these paired helical filaments that then aggregate into masses within the neurons. And so these form these neurofibrillary tangles. There are also mouse models of this, as you'd probably imagine, and they also don't fully recapitulate sort of Alzheimer's type symptomology. This slide just kind of shows like what is kind of summarizes what we know about the role of tau. So its purpose is just to stabilize microtubules. And so when it gets hyperphosphorylated, though, it dissociates from those microtubules and forms its little tangle and neurofibrillary tangle. The microtubules themselves also then become disorganized because they're losing one of their regulatory components. And microtubules are essential for neuronal function because they're important in intracellular transport. And in many cases, they can be, pretty long. They're dynamically regulated, and for example, they're one of they play important roles in basically shipping different components of the cell to the place they need to be, and so which includes transport all the way down axons, for example, which means like it's a relatively long distance for one little cell to sort of try and transport something. So if you start messing with those transport processes, you're likely to cause like major issues with neurons. And the point of this slide isn't to confuse you, it's just to sort of kind of give a sense of how we have all of this information about factors that are involved in Alzheimer's disease, but nothing is really, at this point, coming together as like one clean, easy to state hypothesis. We know that we have those toxic fragments of amyloid beta 42 is capable of causing some problems with neurons. We know that problems with tau can cause problems with microtubules, intracellular transport, and neuronal function. And so you can put those into sort of transgenic models, but they don't give you the full all of the features that you see in Alzheimer's disease. And it may, of course, simply be an issue with that, like maybe mice aren't very good models for Alzheimer's disease. Yes? Yes, so all patients with Alzheimer's will have the cholinergic deficit. They'll all have amyloid plaques, and they'll all have neurofibrillary tangles. And obviously, in general, you can only assess that in deceased patients, but now there's actually the ability to image amyloid plaques in living patients. And so you can actually look at the relationship between buildup of those plaques and disease progression. So what about a different type of hypothesis? There's actually a link between glucose metabolism and Alzheimer's disease. And this basically boils down to the idea that the brain is actually one of the most metabolically active organs that we have. And because of that, it obviously needs a steady supply of glucose. And as we've talked about, if neurons sort of run out of glucose, they obviously then run out of ATP, and then they will, if it's a sort of a nice slow and steady starvation, then they'll undergo apoptosis. You know, like if it's sort of really abrupt, then they'll do swelling and bursting and things like that. So in Alzheimer's patients, you actually see a reduced, compared to age-matched controls, you see a reduced utilization of glucose in the patients before they become symptomatic. And of course, that obviously raises the question of how do they know these people are going to become Alzheimer's patients? And of course, they don't. These are from longitudinal studies where they have like huge populations, and they're just doing these studies. And then 10 years down the line, the patients that are in this gigantic longitudinal study turn out to develop Alzheimer's disease, and then they can go back and say, we saw some changes before the disease appeared. So we see this decreased utilization of glucose in Alzheimer's patients prior to the development of Alzheimer's. And this, the utilization of glucose declines in pretty much this, with a very good tracking of the development of the dementia in these patients. And it's particularly noticeable in certain brain areas, which are listed here, but you don't have to worry about remembering. Oops, excuse me. And those are also areas that are associated with some of the other pathological features of Alzheimer's, like the accumulation of the sort of particularly dense accumulation of plaques and tangles and so on. You also see some of these changes in patients who carry things like the ApoE4 gene, but do not appear to be developing Alzheimer's disease. And so this is how this might work, is kind of summarized here. So obviously, the brain requires a lot of glucose. If we're reducing glucose transport into the brain, then the metabolic load that neurons in particular regions can support is going to be reduced. So some of those neurons are going to run out of energy. That will then lead to sort of the underlying processes that will trigger neuronal apoptosis. And they can also cause oxidative stress, which can initiate protein misfolding and protein aggregates. And in this way, we'll get the features, pathological features of Alzheimer's disease and neuronal loss. And so this theory does seem to sort of have some weight behind it. So we have another chance to talk about where we've got so far.
There's a decrease in acetylcholine, acetylcholine, your brain is not as much. Yeah, so that it shows that parts of the brain. Yeah, so like the acetylcholine like makes you go less. Amyloid hypothesis. Wait. So yeah, amyloid hypothesis. Yeah. Induces wakefulness and pregnancy.
That's a very good analogy.
Okay, so do you want to be in charge of the? Yep. Yes. So that's why the ApoE4 one said an aside. So it's has a strong genetic association with Alzheimer's disease, but no one really understands like why cholesterol transport is going to be, like what the link is between cholesterol transport and the disease pathogenesis. Yes. Yeah.
So, I mean, they haven't figured out exactly why.
Yes, exactly. So we have these theories that kind of explain aspects of the disease without having like some clear unified theory for what's going on. And so we're just going to finish up touching on what we might do for patients who have Alzheimer's disease, because it would obviously be wonderful if you could prevent it or halt its progression. But we're not there. But they do have a treatment, as I mentioned, that is given to Alzheimer's patients and actually slows the progression of the disease. And that treatment is based on this sort of observation of the loss of these cholinergic projections. And so if you have a weakness in neurotransmission using acetylcholine in the brain, what would you use? It sounds kind of like the weakness in acetylcholine mediated neurotransmission that you see in those myasthenic disorders, right? So you actually use essentially the same drug. You have to use, obviously, we're trying to do it in the central nervous system now, so you have to make sure it's a drug that can pass the blood-brain barrier. But there are inhibitors of acetylcholinesterase that actually will enter the brain. And in the brain, they do exactly the same as they do at the neuromuscular junction for these sort of myasthenia gravis patients. And so they just kind of help support cholinergic neurotransmission. And so that's why they don't do anything about the sort of like progressive loss of neurons, but they do help fight the development of the dementia. And they slow that progression. What else might there be? So there's been approaches to look at vaccination against the amyloid beta 42 protein, that toxic fragment. And that seems to have some promise in mouse models. There's some evidence that use of NSAIDs can help reduce neural inflammation, and that can helps provide some level of protection. Again, that's from those large-scale longitudinal studies where someone might be having long-term NSAIDs for some other reason. Because of this glucose metabolism hypothesis, there's some thought that there might be, people have noticed that there's an association between diabetes and Alzheimer's disease. And so that's the subject of investigation. And then, as I think I mentioned, there's a large-scale phase 3 trial for a drug, aducanumab, which actually, goes into the brain to sort of try and break up amyloid plaques. And so this is in phase 3 trials, which is 2015 to 2020. So there's not really any data out there yet. But in 2022, we hope to hear something about it. And then at that point, if, for example, this really is a magic cure for Alzheimer's disease, then we'll be very certain that the amyloid hypothesis is actually correct. This is the sort of fundamental, process that occurs in the brains of Alzheimer's patients. If it's obviously a failure, then we'll at least know that that in itself isn't the answer, because the phase 1 and 2 trials for this have shown that there is a reduction in the buildup of the plaques in these patients. And so tomorrow we'll be talking about Parkinson's disease and Huntington's disease. So continuing this nice cheery bunch of lectures.